Okay, so now we're going to see what happens if there is some sort of an error either in the copying of the DNA during DNA replication or in the manufacture of messenger RNA from DNA. In either of those events, if we have a mispairing of complementary pairs, we have created what is called a mutation. So a mutation is any time we have a permanent change in the DNA sequence. Sometimes it happens with messenger RNA, and that isn't as big a deal. It's mostly the most important changes are those that are permanent, the ones that occur in the DNA. Now, mutations are what ultimately give rise to cancer. And it's easy to see when you look at the DNA of individuals with cancer, you can see that they actually have cells, the cancerous cells have accumulated multiple mutations in their DNA. It's not just one. You actually need to have anywhere from 4 to 6 mutations occur in DNA before you run the risk of actually having some sort of a cancerous cell arise. But mutations can cause cancer, and for that reason we could say that mutations are carcinogenic. Genesis means to generate, right? And carcino means relating to cancer. So a carcinogenic agent is one that gives rise to cancer. That's different from a mutagen. A mutagen is simply an agent, maybe it's a chemical, maybe it's ultraviolet radiation, whatever it is, that causes a mutation. But just because you cause a mutation doesn't necessarily mean it can lead to cancer. Remember that DNA has a lot of intervening sequences of genes with non-coding DNA in between. If the mutation occurs in this region right here where there's no coding DNA, then that mutation doesn't matter. It's not going to give rise to cancer. It only is it only has potential to give rise to cancer if the mutation occurs in the region of a gene. Okay, so carcinogens cause cancer, mutagens only cause mutations. Now, there are two different types of mutations. There are what are called point mutations, and there are what are called frame shift mutations. So let's go ahead and take a look at these. By the way, this picture off to the side here is just showing you how cancer might progress. We are going to talk more about cancer in a little while, not necessarily later in this lecture, but in another lecture, and we will return to the topic of cancer, albeit briefly. So let's look at the two different types of mutations, point and frame shift mutations. In point mutations, we change the nucleotide at a single point along the DNA chain. So if this is my original DNA right here, ATG for example, then what happens is if I have a point mutation, I could have maybe a point mutation so that the G is accidentally copied as an A. It could be copied as a C. Maybe instead at the beginning of the codon I change that G. Any of these are called point mutations, and you will notice that when we undergo transcription, that means when we undergo the manufacture of messenger RNA, that ultimately affects the messenger RNA sequence and makes it different from what the original sequence was. And that simply means then that we have the possibility of coding for something other than what we wanted to. Notice that in the original sequence here, we coded for the amino acid tyrosine. In this point mutation, which happened in the third position of the codon, we changed this to an A. That led to having a messenger RNA sequence of UAU. Remember I told you that the codon sequences, that there's some redundancy in it, that multiple different sets of triplets can sometimes code for the same amino acid. That's called redundancy. Well, here's an example. It doesn't matter whether I had UAC or UAU, both of them still coded for tyrosine. So in this case, this particular point mutation is really a silent point mutation because it did not result in any change at all to the amino acid sequence of the protein. On the other hand, if we had changed this to a C accidentally, now we have UAG as our messenger RNA, and it turns out UAG is one of the 3 stop codons. So this would cause a very serious point mutation that would cause us to stop the manufacture of the polypeptide chain at that codon, and it would not be able to elongate any further. It would be be stopped prematurely. Compare that to the last one here. In this case, now we're doing a first position point mutation. It's in the first position of the codon, and that change also results in a different sequence in the messenger RNA, which ultimately leads to a different amino acid being coded for, and this is going to cause us to have a protein that does not function correctly. I'll give you a little hint or a little secret here. If a point mutation occurs in the third position of the codon, you have a fair chance that it's not going to cause any change in the amino acid that's ultimately coded for. However, if the change occurs in the first or the second position, you're almost always going to get a different amino acid coded for. And if you don't believe me, go back to that amino acid chart that we looked at previously, and you'll see that, for example, in this column here with arginine, notice they are all CG. It doesn't matter what's in the third position, as long as you have C and then G in positions 1 and 2, you're going to get arginine. But if I were to change the first position to A, I won't get arginine anymore. I'd get serine now. So you can see that that redundancy allows for some wiggle room. Okay? It means that sometimes I can have a mutation occur and still be able to make a totally functional normal protein in the face of that mutation. Point mutations are one type of mutation. A more serious type of mutation is called a frame shift mutation. And in a frame shift mutation, I end up either adding or deleting one of the nucleotides from the DNA. So the end result of that is that it completely shifts how we read the triplet codes, and that means it completely messes up the polypeptide chain we produce. So I'll give you an example. Imagine that this is my DNA chain. It's actually just a sentence. It says the big fly had one red eye. It's nice because it's all words that are only 3 letters long, so it fits our kind of our codon idea. If I did a point mutation and did, say, the bit fly had one red eye, the worst that might happen is that I might get a single amino acid change there, or I might get a termination code or something like that. But let's look at what would happen if I added in, for example, a T right between the I and the G here. So here I've added in a T. I haven't substituted it for something else. I've just added in a whole new T that never was there in the first place. And you will see now that look at what's happened to our reading frames. I no longer have triplets that make sense. Okay, and as a result, I'm going to code for completely different amino acids than I did before. The same thing would occur if I'd completely eliminated the G in big. Now the F shifts over to the third position, and again we get this nonsensical triplet codes as a result of the deletion. And I hope you can see from this that a frame shift mutation is far more dangerous, or what we say deleterious, that means damaging, than is much more damaging or deleterious than is a point mutation.